Okay, well, thanks everybody for coming. Those of you that looked at the schedule for the World Soybean Research Conference noticed that there was a SoyBase workshop, and it was 2 hours long. Michelle and Jamie asked me to give this talk, and I said, okay, well, I got 2 hours worth of stuff. And they said, you can have 20 minutes. So I'm going to talk really, really fast, and we'll try to get through it. SoyBase, this is what I'm going to talk about now is this is what happens after you guys do your stuff. You do research, publish results, and the community sees it in the paper, the publication that you put out. They may or may not see it, but most of them get seen. But they're standalone. Basically, it's an island at that point. What we try to do is to provide an environment so that everybody's data, everybody's results can be put into one place, correlated, and then you can start asking questions across experiments, across labs, across even data types to try to learn something that would be difficult to learn by reading any one paper. So, recent additions. I'm going to show you some of the new things that we put in the last year or so, and this is the group of people that have contributed to the stuff I'm going to talk about today. And that's what we're going to talk about, those 6 things. I have a hexafecta. I mean, we're moving up in the world here, right? Nobody's going to beat 6. Okay, I have trouble seeing what's going on here. GEO is a place, as you know, where gene expression data can be deposited. It's a central repository. Each individual experiment goes in as a separate set of records. They're more or less annotated at GEO. There's not very much of standardization there. So what we've done, let me stop just for 1 second. When I say we, what I mean is the group. If you like what's going on, then it was me. If you don't, then I'll tell you who's responsible for this stuff. What we've done is collect all of the data that we could from GEO, where the expression data could be correlated with, could be matched up with specific gene models in soybean. So if somebody does an experiment using private data, a private chip or whatever, and we can't figure out which gene it belongs to, it's just not in SoyBase, because there's no way to present those data in any way that we have. So we have this is the front page going into SoyBase. Yeah, maybe the red button. Yeah, up in the menu under expression, there's the GEO link. You come into this page, and it shows you all the experiments that we've picked up data for so far. We have 2 kinds of data, what we call descriptive data and comparative. Descriptive is just that. If you've done an atlas, you looked at different tissues, just where you've looked at no treatments, those are descriptive. Comparatives where there's some kind of treatment going on. There's a table of contents at the top. Down at the bottom, we have individual experiments listed. Run the cursor over any one of these, and you get a small pop-up that tells you what that experiment is about. And you can mix and match and select the particular experiments that you care about, any subset of these. We also, because you might not care by experiment, you might actually care by tissue. We've also organized these data by tissue. So you can say, I only want to see just the experiments that had something to do with leaflets. So you can again pick subsets of these that you want. As you select the things that you want, the next step, when you hit the proceed to next step button, is to show you what you've asked for. So now what you see here is every sample for that experiment is shown here. But it may well be that, for reasons that are obscure, you really only care about things that happened at 24 hours. So you just uncheck the ones you don't want to see. So those will be invisible as you go further on in the process. So you can mix and match. If you picked up a whole bunch of experiments having to do with leaflets, and all you really care about is the untreated version, you could then just uncheck all of the treated ones, and then you've now selected all of the untreated leaflets across all experiments. So it allows you to subset by sample, by experiment, and by tissue in ways that would be difficult to do with any other tool. Hit the next button. Here's what you've asked for. This is just your final summary. This is what you're going to see further on. What's been going on is that we've been accumulating all of these data into what we call a shopping cart. At this point, you can say, well, okay, I've got all the leaflet things I care about, but I also care about the root ones. So you can now go back, hit this, add another experiment or tissue, and that just drops you back to the front page again. And now you can pick another set of experiments for other reasons, accumulate them the way we just went through into the shopping cart, and end up at this page again. Once you're happy with all of this, you go to the genome browser, and we show you the standard SoyBase genome browser with the expression values underneath each one of the gene models. And I'll zoom in a minute to show you what it looks like. There's always a red question mark on every gene, on every track in our genome browser that pops up and tells you something about the experiment. So what do the colors mean? What are just basically it tells you about the experiment and gives you the ability to download those data if you ever wanted to. So there's a whole chromosome, I think, at least a big fraction of a chromosome, for a particular experiment where all of the different samples are shown here. And of course you can zoom in. In this point I'm showing, the other one was a gene chip experiment. Here's a case where I'm showing you an RNA-seq experiment where we're zoomed way in. There's the gene models, gene model track, and here's the expression of those genes in the different tissues that were tested in that experiment. These are all normalized, and again, clicking on the red question mark gives you the information about how we normalized it and how to interpret what's there. So this is a way of getting expression information, expression data for your favorite gene or a particular region, or however you want to subset these things, you can do this. And of course, I'm only showing one experiment here, but you can choose as many as you want. So if you wanted to compare across experiments, you can do that too. There's no upper limit in what will allow you to do, but you'll get very, very tired waiting for a lot of experiments to show up at one time. It just takes a long time to generate these pictures. So I would suggest that you be judicious about how many experiments you try to do at one time and how big a region you decide to look at. But if you're very patient, you can do anything you want to. Okay, using a similar interface that I showed you a minute ago for the experiment picker or the tissue picker, we also have all of the methylation data that we've been able to get our hands on so far. You can do the same sort of thing, the same sort of pages you end up with, again in GWAS, with the gene models and the genome methylation, showing CGs, CHH, and CHGs. So those are separated out. And the up and down from the line shows you which strand was being methylated, the C in which strand was being methylated. And of course, again, the red question mark tells you about what you're looking at. So now you can start seeing which genes are methylated, which genes are not, which regions around genes are methylated. You begin to ask those sorts of questions. And of course, what's really interesting is to put, well, there's actually a zoomed in version where you can start getting a lot of detail. And then this bottom track, something which is not often shown, is the sequencing depth for that experiment. So you can see here's a case where there just weren't any reads in that region. So the fact that there's no methylation is uninformative. So knowing the read depth helps you interpret what you see in terms of methylation. So there's a zoomed in methylation, but of course there's the fun one, where you put the gene models, you put the genome methylation, and you put the gene expression. And now you can begin to start visually asking questions. Is methylation related to expression? Could it be? And if it is, what kind of methylation could be important? So again, this is a browsing tool, but it allows you to see summarized a lot of experiments, a lot of data, all in one place. Okay, that's gene expression and methylation. Totally changing subject. The pedigree data for soybean is scattered around in a lot of places. It's not trivial to find. What we've done is, as much as possible, is to take all of the cultivars that have been in the uniform tests any time since the beginning of time, since they started doing those things, and then traced their pedigrees back using the literature as far back as possible. And so this is the front page, and there's where you find it. It's under Tools in the Soybean Pedigree Database. This is a list of every cultivar that has been in the uniform trials or anything in the pedigrees of anything that's been in the uniform trials. Is all listed here. Now, if you're looking for a particular one, of course, you can just search for it, You can also do various filters, or else you can just scroll through the whole thing if you want, although it would take a while to scroll through. But if you pick one, in this case, we picked Bragg, the page you get tells you there's what you ask for, there's the things the name that it's also known by, there's its pedigrees, and then here's all of the records that contain Bragg. That is, it was in a cross that generated another line. It was somewhere in the pedigree of that line. And you can begin to track those down. You can see why Bragg is there, what this line was derived from that cross. So again, you get to see the history of the Bragg cultivar. One thing I didn't mention is that SoyBase, any text that's in green is a link. It's a clickable link. So in this case, if we ask for Bragg, if you then click on this COCR 485, you'd get an equivalent page, but it'd be for COCR 485. So it allows you to walk quickly through the pedigree and see who's related to who. There may be particular lines you care about. And this button here does exactly what you might think it does. It goes off to Google Scholar and finds every reference it can to Bragg. And it just returns something back like that. We take into account all of the alternative names when we do the search, so that no matter what it's called in the publication, if it's the same line, you'll find the same information. So that's pedigrees. Number 3, GWAS QTL. SoyBase has for the last 25 years had QTL information in it, biparental QTL. That's one of the strong points of the database. GWAS QTL is a new technology. All of you are familiar with it. Enough of those papers have been published now that we've been able to begin to put those data into SoyBase. The displays are slightly different than what you're used to seeing, but it'll be pretty clear once you look at it. This is an example for the GWAS report page for plant height, what we call plant height 1. Now that 1 simply means that it was the first paper that had a GWAS QTL for plant height that we put into SoyBase. So it has nothing to do with timing or alphabetical order or anything else. It's the first one we ran into. The nomenclature that we use for GWAS QTL is the same that we use for biparental QTL, by which I mean that the text at the beginning describes the trait being measured. There's a number dash number. The first number is just something to be unique. It's an integer that represents a particular paper. That's all it does. And the information after the dash is a counter for the number of QTL for that trait in that paper. So if there are 3 QTL for plant height in this particular paper, we'd have 1-1, 1-2, and 1-3. That's the standard nomenclature. For GWAS, we used exactly the same nomenclature, save only that after the dash we put a lowercase g. So that if you see 1-G1, you know it's a GWAS QTL. 1-1 is a biparental QTL, but they could be for the same trait. A link to the paper, ontology terms, and here's the QTL truncated, but here's the QTL list for all the QTL for that trait from that paper with a few summary statistics and links out to the genome browser and to the genetic maps. There's the genetic map display. This is going to become one of the 2 standard genetic map displays at SoyBase. The one you're used to seeing has biparental QTL on the right and another genetic map on the left. And what we're doing is switching it over to having the biparental QTL on the right, GWAS QTL on the left. Same organization, same nomenclature, same colors, same columns. So you can begin to compare these exactly. So things which are in blue on the left are the same type of QTL as the blue on the right. If they're in column 2, then that tells you something. The 4 columns just help subdivide the QTL into yield-related, disease-related, plant structure-related, and everything else. Those are the 4 columns that we use right now. So this gives you a quick way of comparing GWAS QTL and biparental QTL in one display. Markers which are in common between the 2 maps are in red, ones which are not are in black. And so this is just a summary way of looking at the data for all the experiments we know about for this particular trait in this particular region, both biparental and GWAS. We can't put biparental QTL on our sequence map for a variety of reasons, but we can put the GWAS QTL. And so we will have GWAS QTL listed in our sequence browser, along with the gene models and other markers and any trait. You can turn any track you want to on and off. So this is a way of saying I found a region I think is interesting. I'm convinced it's real because there are both GWAS and biparental QTL for that trait in that region, so it's probably not an artifact. What genes could be involved? Well, there's the QTL, and there are the genes, and you can begin to track things down that way as a visual browsing tool. Okay, new subject, number 4. We have a lot of data in SoyBase, and in many cases we present it to you in a way that's what we think is the very, very best possible way to do it. However, not being perfect, there might be other uses for these data also. So we make most of the data that we have is available as download one way or another. So from the front page, we have a whole section of our toolbox for data download. If you click on that link, you go to a page that looks like this. We have genetic-based data downloads and all sorts of sequence-based data downloads. Almost all of these are there because somebody asked me for a particular data subset or something they wanted from SoyBase and they couldn't figure out how to get it. Well, we can usually figure out anything. So we got the data and gave it to them. And then thinking that if one person asked, a dozen people didn't, anything that gets asked for, we make into a tool and put onto this page. So these are the ones that we have right now. But if there's a subset of the data or something that you like from SoyBase and you can't figure out how to get it, just ask. First, we'll give it to you. And second, we'll put it onto this page so the next person will have an opportunity to use that same tool. So taking it as an example, one of the new ones we've added just recently, SoyBase has a very extensive BLAST tool. We have a lot of targets, genomic, genes, the organelle DNAs, all sorts of things. In fact, there's a list of all of the targets that we have for BLAST right now. When you go to this page, they're all listed. If you pick one of these, you notice they're all green, so these are all links. If you pick one of these, I've just picked the second one just because it all fits on a page, drops down to here, you have the opportunity to do your BLAST against that target, this is just one way of doing that. But what I want to point out here is that the opportunity here is to download all of the data that represent that BLAST target. So in this case, what you'd get is a FASTA file of all of the transcript sequences from Williams 82 A2.A1. So if you wanted all of the gene sequences from soybean, you could find those by going to NCBI with some effort. We've gone to the effort for you. So it's available for you right here. And all of these are available as FASTA downloads. So this will just save people a little bit of time, at least we hope it will, rather than trying to generate that for yourself. We try to keep these up to date. The BLAST targets are reasonably up to date. And as we get new data, if it's a new type, it'll appear in this table of contents as another line. If it's just updated data, it'll just be up to date. Whatever you ask for here, you'll get the updated data. Okay. Now this is something which is near and dear to my heart. You guys are far too productive. Okay? If you just take a year off, we'd have some hope of getting caught up. But since I don't expect that to happen, we're working like crazy just to barely keep up with the community on a small subset of the data types that could go into SoyBase. Congress is unlikely to give us another team, and so we have what we have. What we've done is we're going to ask you to help us. We've developed 3 so far spreadsheets for data entry. That is, these are relatively simple spreadsheets that represent the data which you've generated for your experiments. The stuff that we've asked for, I don't have any examples here, but I'd encourage you to download these spreadsheets and look at them. It is the same data that you used for your publication. We're not asking for anything that's not there. But it's organized in such a way that we can relatively quickly get it into SoyBase. So if you publish a paper, it may take a year before it shows up in SoyBase, just because of the limited manpower that we have. If you give us this spreadsheet, it'll show up right away. So if you want to get your stuff in more quickly, this is the way to do it. If you don't or can't, well then it'll get in when we can put it in. We don't ever act as editors. Anything that's published, we will attempt to get into SoyBase, but it may take a while. So I encourage you to use these tools as much as you can. For some of the data, there's not good formats yet. So we don't have spreadsheets that are obvious about how to use yet. So what we have here, of course, is Ask Us. If you've got any kind of data that's not one of the standard ones here at the top that we have templates for, we still want to have those data. Get in touch with us, and we'd be glad to work with you and try to get it in. And then this bottom paragraph is one that is really, really important. We spend a large fraction of our data curation time is spent on what we call data forensics. That is, the publication gives us 90% of what we need to be able to put it into SoyBase. Or sometimes it gives us 10% of what we need to put it into SoyBase. Nomenclature really matters. I mean, lots of things matter. But it would really, really, really help when you start a new experiment that you know the data are going to go into SoyBase. Talk to us at the beginning of the project. We have a lot of experience with this, and we can help you do the experiment. We're not going to help you design the experiment. That's not our job. But we can help you collect the data and present the data in such a way that it can rapidly be exposed to the community in a useful format. The end result of your research should not be the publication. That should be the end result should be those data showing up in SoyBase in the context of what everybody else has done. And it would really help us if we can do that, if you guys can work with us to do that. There's the Contact Us. We have a page. You can put any kind of free text in here. It will get a response. They come to me. They come to all of the team. But they come to me, and you will get a response. And there's the end. I've also been told that my time is almost up. But I'm here in the building. I'm always around. If the door's open and I'm not on the phone, I'm interruptible. That is, I'm just not doing anything that's so important that you can't knock on the door and walk in. If the door's closed, I'm not in the building, but I usually come back. At least so far I've come back. They keep paying me, so I keep coming back. I'm down in G304, email address there, phone number. You're always welcome to walk in anytime. And I'd be glad to talk about SoyBase or soybean or almost anything other than baseball. I don't know anything about baseball, but other stuff we can talk about. So with that, that's all my time, and it's also on my slides.
When was it started, SoyBase? Was it early 90s?
SoyBase started about the end of 93, beginning of 94, on that order. I joined the group in the summer of 95.
So it's going to be 25 years? Okay.
Yeah, on that order, right. I've been here 22 years, and so it's about a year older than me from my career here.
Is there any celebration going to be coming up next year? Maybe.
Maybe. I may not be employed next year. Yeah.
I have a very specific question. So when you say you normalize like the RNA-seq data,
Mm-hmm.
you're normalizing the entire data set, not just what's being compared? So like whatever that you have to preload, you've done the normalization up front.
Yes.
And then the user selects what tissues they're interested in.
That's correct.
Have you gone back to look at the effect of your normalization on like papers from where you know what the differential gene expression is?
We are currently only accepting experiments that they did some sort of normalization on the experiment. We do not normalize across experiments.
Okay, that would.
So
yeah.
all you can really see in those pictures is the trend between experiments. Like it's high in all the experiments, well, it's probably high. If it's high here and low here, I don't know what to tell you. But we don't have the manpower to take all all of those reads and
Yeah,
renormalize.
I just wasn't sure.
Yeah, no, it's normalized within an experiment, and the color coding is within a track. So the samples may not have been sampled equally depth, but within a track, the 5 colors represent the relative expression.